Today we are going to explain about synthesis of antibodies. We have been explaining antibodies throughout the lecture from topic 1 to 5, and today we are going to explain how the body or the organs of the immune system use in the synthesis to follow the step-by-step mechanism in the production of antibody in vivo within the body. Now, antibodies synthesis is just a process by which immune cell creates specific proteins. Mind you, the antibodies, they are just protein to neutralize invaders like the bacteria, viruses, or all the antigens that you can name. The process primarily take place in a specialized blood cells called the B cells, which are the bone marrow cells, because they are produced in the bone marrow and matured in the bone marrow. Now, before an antibody is produced in the body, there are certain trigger mechanisms. The body have to notice certain situation, then it will trigger the step-by-step production or mechanism for the production of these antibodies. So the first trigger point is that the B cell binding must bind to a part of an antigen called the epitope. So after antigen presentation, the B cell have to bind to the epitope. So when it did that, it's going to internalize the B cell, swallow the antigen receptor, so it will via a process called endocytosis. After endocytosis, the body will present the epitope of the antigen on the surface of the B cell, so that the B cell can be recognized by the epitope on the surface of the B cell. So every B cell in your body has a unique B cell receptor site, and that B cell receptor site is called parotype. When a foreign substances and antigen enters the human body, it eventually bumps into a B cell with a receptor that fits it like a key and lock. So from this side, you can see a depiction. These red parts are B cells receptor site that they can bind with the antigen before internalization, then presentation for the continuation of the process. Now, after the trigger, the B cell have to be activated in the second stage. The B cell need a co-signature from another immune cell to start the production. This prevents the body from accidentally attacking its own tissues. So in the step 3 is called the presentation. The B cell break down the antigen and display the fragment of the antigen on the surface using a protein called the major histocompatibility complex, which stands for MHC2. So the presence of the presentation of a fragment of the DNA is called opsonization. So it will be presented using the major histocompatibility complex. Histocompatibility means cell by cell or tissue by tissue compatibility coming together to present the fragment of the antigen or the epitope. Now at stage 4, the T helper cell recognize this fragment. So it's like you see someone with the shape of something on the surface of it, then you will recognize that there is something within the body. So the T cell recognize the fragment on the surface of the B cell, then it will attract the action of the cytokines, which I tell you they are working by concentration gradient. These signals give the B cell the green light to begin multiplication. Then immediately this happens, the B cell will enter into what is called the clonal expansion and differentiation. In clonal expansion and differentiation, the single B cell divides rapidly, creating thousands of identical copies of clonal expansion. These cells then differentiated into two main types. The memory B cell, these stay in the body for years, usually located in the spleen and the lymph node, where they are sorted and reassorted there to provide immunity if the same germ or antigen enters the body again. Then the plasma B cells, these are the antibodies factories. So you have an antigen, it enters the body, it is recognized by the available B cell, then it will present itself using histocompatibility complex. This will trigger the action of the T helper cell, and T helper cell using cytokine will trigger the functions, the differentiation and clonal expansion of the B cell. So the B cells produce memory cells at the end, then plasma B cell. Plasma B cells are just stem cells that they are used for the production of other memory cells against future invasion. So that's why they are called the antigen or antibody factory. Antibody factories. That's a company for the production of the antibodies within the human body. And their sole purpose is to synthesize secret massive amount of soluble antibodies. So genetically, this is happening using what is called the VDJ recombination, which we are going to explain now. So, but for us to explain that, you have to know that if you recall back to the structure of the antibody, we have the Y shape, which has the heavy chain, the disulfide bond, the constant region in the heavy chain, and the other regions. Now, the variable region is where there is the Y shape of the antibody, and which is also has the diversity region. The joining region is where there is the disulfide bond. Then there is the constant region, which has the FC region from the heavy chain. Now, collectively, three different types of chromosomes within the gene are coding for this part. The heavy chain, which has a collective of genes with 14 chromosomes. Then the light chain, which has 2 chromosomes, and the light chain gene with the lambda have 22 chromosomes. So collectively, more than 38 chromosomes code for the production of one antibody within the DNA of that particular plasma B cell. But for us to clearly understand that, we have to follow what is called somatic recombination, or more simply VDJ recombination, where V stands for the variable region, D stands for the diversity region, and J stands for the joining region, which is the disulfide bond. So during the cell development in the bone marrow, antibody genes undergo somatic recombination, a process that randomly joins the genes segment together to form the VDJ complex, which by translation and transcription, it will lead to the formation of the fragment of the Y-shaped immunoglobulin. So the heavy chain arrangement, one VDJ segment combines, and it followed by association with the C region, which is the constant region. This will give you the heavy chain region of the antibody. Then in the light chain rearrangement, one V and J region standing for variable region and joining region will come together to give you the variable region, which is the Y shape region. Then there is no D segment, which is diversity region. Now the rearrangement is mediated by what is called the RAC1 and RAC2 enzyme, recombination activating gene. This recombination activating gene, they landed in a landing pad. They landed in a landing pad which is called the activation signal sequence. This reactivation signal sequence, they go into the gene and capture the variable region, the diversity region, and the join region, and they will merge them together. So when they merge them together, they will become one continuous and complete gene. But they did this by following, they did this using a Planck sequences that contains the VDG segment. So first of all the RAC1 and 2 find 2 segments it want to join together, it brings them together and physically snap the DNA backbone. So when the VDJ region are combined, the remaining aspect of the gene is snapped together. It will be cut alone. Then it will be hairpinned. It's like a lady collectively tighten her hair. they have a pin that they use in tightening the hair to close the end of the hair. So exactly what is happening to the DNA when these segments are collected together, a hairpin will be inserted to tie them together so that they cannot escape or removed from the combination of the genes. Now, the processing and junction diversity, the enzyme called the Artemis enzyme open the hairpin. When it is jammed together, now it is ready. Then the Artemis enzyme will open the hairpin. Then another enzyme called the terminal deoxynucleotide transferase randomly transfer random nucleotide or random sequence. This is used to make sure that each individual antibody is uniquely formed and is not the same with the other antibodies. So that's why we are using the TDT enzyme. This ensures variability of the produced antibody after synthesis. Then ligation. Ligation is just tightening the end together. Since you already have the sequences of DNA, then you tie the end together. And this is done using non-homologous end joining. This is just a machinery which ordinary DNA is used in correcting sequences of DNA. Now in this situation, since you tightened an unspecific and random nucleotide at the end of the hairpin, then you have to use non-homologous sequence typing machinery to make sure that it is glued and the VDJ component of the segment is not damaged. So when you did it, you finish with that. But most importantly, now you have your antibodies after you combine all the action of the RAG1 and 2, the recombination signal sequence, the use of the Artemis, the use of the TDT enzyme and the non-homologous sequence joining. You all have that. At the end, you have your antibody. Now, for you to test whether the antibody they are working effectively or not, you have to test them against safe antigens within the bone marrow. And this process collectively is called negative selection. So if the antibody produced binds strongly with safe antigen, meaning it's very dangerous to the body. So it can bind strongly with the safe antigen. So when it did that, then the body will invoke the negative selection occur in the bone marrow. So the body will enter into what is called the PET of the antibodies. PET of the antibodies after negative selection. So in PET 1, which is a success no reactivity, if the antibody do not bind to the self antibodies, then it will allow to pass and go to the spleen or the lymph node, where it will be sorted and wait for future invasion. PET 2, if the receptor ending, it is retagged again. That means if the antibody produced bind to self antigens, then the whole process will start again. It will invoke the action of the RAG1 and 2, the RSS, the Artemis, the TDT, and the non-homologous end joining, so that the antibodies will be entirely reproduced again. Then PET-C is a situation where the antibody will just enter into death sentence, allowing it to commit its suicide by itself. So certain enzymes within the antibody will be produced and it will enter into a situation called apoptosis or destruction of itself. Then FET-D. FET-D is called an energy or dormancy. Those that are weakly binded to self-antigen are allowed to enter into energy. Energy is just a sleeping and they will never wake up. Then peripheral tolerance. Now nothing is perfect. The fact that even there is sorting and making sure that all the antibodies produced in the bone marrow, they enter into peripheral blood circulation without any problem, there is still a tendency and probability that some of them, they can bind to the self-antigen, but they can escape the bone marrow and enter into peripheral blood circulation. So even if they did this, already nature has made it possible in such a way that there are certain complex substances or cells that they can police the peripheral blood circulation to make sure that the antibodies produced are functioning effectively. And this can happen through the following. First of all, it's called the clonal energy. It's a situation if a B cell binds to self-antigen in the peripheral blood circulation without receiving a co-stimulatory signal from a T helper cell, it becomes an anergic, which is the dormancy I was telling you. It will enter into permanent sleep forever. Then number two is a situation where the B cell requires permission from T helper cell to produce antibodies. Then there is another situation with the use of regulatory T cell and regulatory B cell. They are called the BRACs. The BRACs and the TRECs, specialized peacemaker cells, release as anti-inflammatory chemicals like interleukin-10 that suppresses self-reactive cells and keep the immune response in check. But despite that, if eventually the negative selection fails and the peripheral policing or peripheral regulation fails, then the body will enter into what is called the autoimmune disease. This is a situation where the immune system loses the ability to distinguish between self and non-self, and this leads to the production of autoantibodies. These autoantibodies are antibodies that they can fight the self-antigens within the body. So they can destroy cells in the body, they can destroy tissues in the body. So the body is producing dangerous substances against itself. So if this is happening, the situation can lead to two diseases. One is called the systemic lupus erythematosus. This is inability to remove dead cells in the body. The fact that you know that the body is generating and also degenerating. So the dead cells can be removed outside the body. But in this situation, the body is attacking itself and the cells cannot be removed outside the body. So it will cause what is called the systemic lupus erythematosus. It's just a situation where the B cell encounter internal self-antigen like DNA or nuclear protein that should never be exposed. So the B cell mistake the DNA for a virus and begin producing anti-nuclear antibody. So people will come down with SLE, the systemic lupus erythematosus. Then number two is rheumatoid arthritis. The B cell in the joint begin producing rheumatoid factors like ACPA, antibodies that attack modified protein. And if they did that, they are going to cause a lot of problem and a very advanced systemic malfunction infection within the body. So collectively, this is how antibodies are produced, how they are how they are checked before they are released into peripheral blood circulation, how they are regulated in the blood, and if it fails, the kind of the disease that they can cause in the human body. So thank you for listening. And next we are going to enter into hybridoma technology, or more simply, production of vaccines.